So now what I'd love to do is show you just how easy it is to run a test. So in our setup, we've got our 2 reagents, our 2 tubes, and our 2 test cartridges. And I have a sample here that will run. So what I'm going to do is I'm going to take this pipette that comes with the system, and I'm going to dispense my sample, and it just so happens to be 30 microliters, into each of the test cups. So 30 microliters in the tube for the white cell test, 30 microliters in the tube for the red cell test. So we have our sample. Now what we're going to do is we're going to layer on our reagents. So we will, just see, I'm switching the location. So I'm going to grab my red cell reagent, and I'm going to add 30 microliters to my sample cup. And because the red cell reagent is an antibody stain, there's a 3-minute incubation at room temperature, so that mixture just sits right in the prep tray. But we're not going to waste any time because while that reagent is incubating, we're going to run our white cell test. So I'm taking 30 microliters of the white cell reagent, mixing it with my sample, and then I'm going to dispense that sample onto my test cartridge. So we dispensed our sample. Now I'm going to take that cartridge over to the vacuum station and pull that stain sample through the test cartridge very fast and move that over to the instrument. And that cartridge just sits right inside the instrument. Now I'm going to go to my computer and I am going to input my patient information. And so here you'd have a barcode scanner that comes with the device. For now, we can key in an accession number. This is the TNC test. We're not going to make a dilution. We've tested this sample neat. Let's say that this is a clear and colorless sample and that the source is tube 1. All of these fields are customizable. We believe strongly that we should adapt to your nomenclature. You shouldn't have to adapt to ours, so you can create any colors, any sources, any appearances that you'd like. And then we're just going to take a picture of that cartridge and see what our TNC result is. So just like that, hopefully you can see the green markers on the screen, which are representing the cells that we've just counted. And we'll go ahead and we'll accept our result. The instrument is prompting us to remove the cartridge from the instrument, which we'll do. And then I just want to go back and take a look at this image for a moment. So here's our image. You see the result here, 14 cells per microliter that gets populated in our database. You'll also see the total count. And this is the time savings that I'm talking about. So you'll see that quickly the instrument counted 207 cells. And the instrument knows exactly the volume that was deposited onto the test cartridge. And so instantly, it does the math for you and provides that result in cells per microliter. Really nice way to eliminate potential calculation errors that can occur with the manual method. GloCyte handles all of that. And a nice way to build traceability. For example, you have your user ID. Right now I'm logged in as admin, but you can set up individual user accounts should you choose. You've got your date and timestamp, definitely something that inspectors check for. And you've got your accession numbers all lined up. At this point now, our incubation for the red cell test is complete. So we can go back to our sample preparation tray and simply deposit that stain sample onto the test cartridge. just like we did for the red cell, or for the white cell test rather. So we'll deposit 30 microliters of that sample, still with the same pipette, just a different tip, onto the test cartridge. We'll move that test cartridge over to the vacuum station, where we'll vacuum the stain sample into the base of the cartridge and move that cartridge over to the instrument. Now let's take a picture just like we did for the white cell test. So here we'll choose patient. When you scan or type your session number, the instrument remembers the other properties that you input. We're not going to do any dilution, so we'll start our RBC test, and let's see how many cells are in this patient sample. There we go, 11 cells per microliter. So really quick, fast test. You can get both RBC and TNC results in under 5 minutes. I'm going to go ahead and remove this and go back to that image again. So now we have both our TNC results at 14 cells per microliter and our red blood cell results at 11 cells per microliter on the screen. So really quick, under 5-minute test time. In terms of reportable range, for this instrument with TNCs, you can go up to 6,500 cells per microliter, and for RBCs, it's 615,000 cells per microliter. And we have linearity down to 0 cells per microliter. So really wide range of samples that you can test to.